Have you ever wondered, plants that grow in temperate or tropical zone, where due to high temperature and high light intensity, causes high photosynthetic rate, high photorespiration and water loss, how plants cope up with the situation? If so happens due to high humidity, stomata in such plants will remain closed, and during photosynthesis, CO2 will be consumed while O2 will be generated. Under closed stomatal conditions, O2 will be trapped inside. Rubisco, the carbon fixation enzyme, happens to have affinity with both CO2 and O2. When Rubisco reacts with oxygen, the process is called photorespiration that is responsible for waste of energy and reduction in sugar synthesis. Thus, to circumvent the photorespiration process, plants such as maize, sugarcane, sorghum, etc. have special or distinctive leaf anatomy called Kranz anatomy. Kranz anatomy is the arrangement of cell where bundle sheath cells surrounds vascular bundles and bundle sheath cells are in turn surrounded by mesophyll cells. Such leaves also have dimorphic chloroplast, that is the occurrence of two distinct forms of chloroplast within the same plant. One for fixing CO2 and the other that allows Rubisco to function at increased CO2 partial pressure. Electron micrograph shows the bundle sheath cell chloroplast is devoid of grana, while mesophyll cells thylakoids are stacked into grana. M.D. Hatch and C.R. Slack in 1970 published Photosynthetic CO2 Fixation Pathways in the journal Annual Review of Plant Physiology. Here he provided evidence for alternate pathway for carbon fixation, since C4 plants first assimilate CO2 to form 4-carbon compound oxaloacetic acid, so we call it C4 cycle. The process begins in the mesophyll cells, the plant's primary photosynthetic cells. Here, the enzyme phosphenyl pyruvate carboxylase, or PEPC, combines carbon dioxide with phosphenyl pyruvate, or PEP, to form a 4-carbon compound known as oxaloacetate. Now this oxaloacetate undergoes conversion into another 4-carbon compound, malate, which then travels to the bundle sheath cells. In bundle sheath cells, malate is decarboxylated or broken down to release carbon dioxide. This carbon dioxide then enters the Calvin cycle, a process you are already familiar with. It's here that carbon dioxide is fixed into a 3-carbon compound, eventually leading to the production of glucose, the energy currency of the plant. The Hatch and Slack pathway reduces the loss of carbon dioxide through a process called photorespiration. Fixation of 1 CO2 requires an average of 5 ATP and 2 NADPH for plant. Though the C4 metabolic cycle consumes more energy than C3 photosynthesis, but it enables plants to continue photosynthesis efficiently under difficult conditions.